Hi everyone, good morning and welcome to the Biophysical Characterization of VLPs using a MiniTEM system webinar. My name is Salome and I'm one of the Vax-Hub Research Fellows. This webinar, as it was mentioned to you on the invitation, will be recorded. So if you have any problems, please contact us and you will be made anonymous. Also, it is important that you keep your microphone and camera off to avoid any connectivity issues. The scope of the webinar will be to show the importance of using a MiniTEM system as an analytical tool and to assess the criteria for the image analysis of the particles that we will be using. At the end of the talk, there will be a Q&A session where I will be asking you either to raise your hand or to type the question on the webinar chat. Or alternative, if you want, you can send comments, feedback and questions to myself or to Anne. Our speaker today is Dr. Adam Dickinson, who is an application scientist at Vironova, and Adam is based in the UK. Dr. Saba will join us later on the Q&A session. A little bit of background about VLPs. So VLPs hold key characteristics that make them an efficient platform for vaccine development. For example, they can be produced in a variety of systems such as yeast, plants, insect, and also cell-free systems. They have highly organized and repetitive structures, which enables the induction of strong cellular and humoral responses. Their size is also very appealing. It ranges from 20 to 200 nanometers, which allows an effective cell penetration. As far as the modification of VLPs is concerned, they can be externally and internally modified to display antigens or epitopes of interest. Of course, it is critical to fully characterize these type of particles to be sure that they meet the required efficacy, safety, and regulatory standards before they reach the market. Some examples of VLPs being produced and used are Gardasil, which is a vaccine against human papillomavirus, and Engerix, which is a vaccine against hepatitis B. From all the characterization that I mentioned, one of the steps, let's say, of this control is the biophysical characterization. And for that, we can use the MiniTEM, which is a robust, rapid and reliable analytical tool, and which offers very appealing features such as a short learning curve. It's very user-friendly, low maintenance required, and no need for special infrastructures, which is a plus if we think about using this type of equipment on developing countries. If we compare this system with the golden standard, the conventional TEM, we can see where the disadvantages, let's say, of the conventional TEM, which the most highlighted ones are the need of very specific infrastructures, which are normally very costly, and a high standard of expertise to use the equipment and to perform all the analysis. In this session, our case study will be the analysis of a VLP candidate for a universal influenza vaccine. This VLP was produced in a cell-free system with a minimal purification step, and its structure is the hepatitis tandem core with influenza antigen displayed. This sample was courtesy of Professor Dan Bracewell group. It's part of Noel Collan PhD. Also, we will be analyzing some TEM images that were kindly provided by Dr. Christy McKellen-Gibson from NIVSC. This image, although they are not VLPs, they have a high level of complexity, which will be useful, very useful for us to visualize, because we can do an analogy of the challenge of doing this type of image and what we can face when doing the image, and how we would understand what is our particle, what is an aggregate debris, or any other issues that are not properly what we want. Without further ado, I will hand over to Adam for the live demo and also for a deep explanation about what we will be seeing today. Adam, I will stop.
Salome, thanks for the introduction and the opportunity to present today. And thank you everyone, and good morning for taking time to join me this morning for the remote demo. So now I'm just going to share my screen, and hopefully that this will come through pretty quickly. Just to give you an introduction of what hopefully we'll be doing today. As Salome just mentioned, we're going to be imaging some VLP candidates for universal influenza vaccine. And today's demo will be slightly different to part one, where we had the full live demo with the sample exchange and discussion of sample prep. So I've just got a couple of bits of information on this slide for those who aren't familiar with TEM and what we're actually going to be looking at today. So today we're actually going to be looking at our samples on a 3mm copper grid. This is what we commonly use with the MiniTEM, and it contains 400 square meshes or holes of around 42 microns. So this is kind of the area that we'll be actually looking today. Hopefully going to be placing a couple of different regions on this grid to see how it varies across the sample. And these grids are carbon coated, so very thinly coated with a stable piece of carbon that's only 10 nanometers, so nice and thin. This is flat and conductive, and also hydrophilic in terms of we can actually apply the sample and they will adhere with that. These samples today have been negatively stained with uranyl acetate, and I've just added some links here for those who aren't familiar with the process, but perhaps you can watch offline. They're short 3-minute videos on our YouTube channel, just to show you the process of glow discharging to prepare the grids and clean them, the actual staining process that takes around 5 minutes, and then also the actual insertion of the sample and removal from the MiniTEM system itself, which is currently being done by my colleague who's currently in Stockholm. So I'm currently in Manchester today, controlling the microscope. So we're hoping technology will hold up for us. And what we'll hopefully be doing today, and just let me know if these slides aren't moving with the animations, but we're actually going to be looking at the grid, setting down some places to image, or waypoint as we call it. We're going to be capturing some images. We're then also going to be hopefully using a type of machine learning called convolutional neural networks to actually detect VLPs. And then hopefully we're actually going to generate some quantitative data and metrics from these samples. And as Salome mentioned, we'll be using VLPs, but also some samples of rotavirus that have been kindly provided with us today. So please bear with me. I'm just going to switch screens to the MiniTEM desktop. Could you please confirm if you can see the microscope software?
We can see it. Yeah.
Great. Thank you very much. Please don't be alarmed that the screen is currently black. The microscope is currently positioned over a grid bar, so essentially a solid piece of copper. If I just move using the mouse here, hopefully you can see this, and I'll try and move as slowly as possible. I'll just move the stage ever so slightly, and then hopefully you'll be able to see the beginnings of the sample. So at the moment, we're zoomed quite far out. We're at a field of view of 23 microns. And I'll just go into the center of this image right here. And again, don't be alarmed that the sample is torn or ripped. I'll go through why this may have happened, how we can avoid this. And what I'd like to just take you through is how we actually control the microscope itself. So one of the steps that we've skipped this morning is that we've actually turned the electron beam on. Now, typically we allow this beam to stabilize for around 10 to 15 minutes. And then we go through a process of optimization in terms of centering the beam to make sure that the beam is on axis and transmitting through the sample perfectly straight. We also make sure that it's free of any astigmatisms, and what that is essentially correcting for any differences between the electromagnetic lenses that actually are present in the MiniTEM. And the MiniTEM has lots of automated features to help users who aren't too familiar with TEM itself. And this little icon here is our auto beam alignment function that can allow this to do all the initial optimization steps for you. I've already done this step just to save time, but just to make you aware that these features are actually present on the system. You may be a little bit confused possibly why it says light objective, but the MiniTEM actually uses a combination of electron and light optics. We actually have a crystalline screen called a YAG screen that transmits the electrical signals into photons of light, which are then picked up by either a 4x or 20x objective, and then the image is then formed on the camera, which is hopefully what you can see in the center of the image. So in terms of control of the microscope, we can control the illumination, and some samples can be a little bit more sensitive to others. And what I observed this morning when I first turned the beam on is that at the automatic illumination settings that the microscope is assigned, it's actually penetrated the actual grid and the carbon, and it's led to a bit of a tear. So what I'll actually be doing today is manually controlling the illumination just to protect the sample so that we can get some nice images this morning. Generally speaking, the auto illumination will work fine, but from time to time again, you may have samples that are a little bit more sensitive than others. In terms of image capture, again, we have automatic features in terms of generating the best contrast and brightness for our images. And then we can also have a live stream of actually what we're seeing today. And that's why you can actually see me moving around the actual stage. We also have some background correction features as well, but the actual image you can see here is pretty clean. There's no need to actually perform background corrections. From time to time, you may have some pixelation over time that you may want to remove. And this feature is here just if you do need to actually do that. So moving over onto the right, we have our histogram here. So we have basically an intensity in pixels of our image. The camera goes from 0 up to 65,000 because it's a 16-bit camera. And these values will vary depending on where I am in the particular image. Over here, we have the actual control of where we are on the grid. This circle represents the entirety of the stage in terms of where we can move, and will also represent where we are up on the actual sample grid. So what I'm actually going to do now is navigate around the grid, try and find a nice section, and hopefully we can then see some VLPs. So over here, we can actually just click anywhere, and the actual stage will automatically move with wherever I've actually clicked on the image. And it's not particularly a bad starting point here. You can see some objects are still quite far out. I will zoom in just a moment. Just move slightly to a fresh region. This one looks quite nice. Staining looks quite even. So just get a little bit central. And what I'll do now is I'll just move through the different fields of view. So I'm actually going to be increasing our magnification as we move through the fields of view. I'll just adjust the illumination as well. Move to 6. And hopefully, it may be quite difficult. I appreciate you looking through a screen, and I'm also looking through 2 screens as myself. You start to see some of these circular objects. These are most likely going to be the VLPs or something similar. So I'm just going to move even closer again, down to 1.5 micron field of view. That corresponds to the width of the image. And definitely we can start to see some VLP-like objects here with a bright edge and dark center. I think the optimal view is going to be around 1 micron. You may have noticed we've actually now switched to the 20x objective. That's great. So we've got some objects that we can clearly see. They're out of focus at the moment, so nothing to worry about. We're going to try and get these into focus. So what the MiniTEM actually has is an autofocus tool or function. So I'm just going to adjust the illumination slightly, make that a little bit brighter, and I'm going to ask the autofocus to run. So what the autofocus will do, it will go in 10 micron steps in the Z-axis, and hopefully what you can see up here, nearly in the top right, is they will be changing in initially 10 micron steps to try and find the best focus. It will then focus on a little bit closer, maybe in 2 micron or 1 micron steps, until it finds its optimal position. And this roughly takes about a minute and a half, depending on how many objects are actually present in the actual image. But we found that this is quite a useful tool, particularly if you want to image multiple regions across this grid. Although the carbon and copper grid is mostly flat, slight differences in the region of microns. So it's best to use an autofocus tool, and then essentially every time you move to a new position, you can get an optimal focus. And my plan today is to set at least 3 to 4 different regions or waypoints. I'm going to be applying autofocus, and hopefully we can acquire somewhere in the region of 40 images to give us a nice representation of what's actually occurring on the sample. Now the movements in the Z-axis seem to have become a bit smaller now. Yes, it seems to be around 1 micron, and you can start to see the outlines, hopefully, of some VLP particles in here. I think it's almost done, and I'll also then introduce some of the additional features that we have to try and find the optimal focus. So this is where the autofocus is actually finished its processes. Now, to our actual eyes, this might not look like it's in focus. It's actually in focus for the microscope, but it's probably not in focus for our eyes and our image analysis algorithms. So typically what I would do here, and this is very similar to what I would typically do for any live cell imaging or bright field imaging, we defocus the sample ever so slightly just to enhance the contrast and the edges of some of these objects that appear to be in the image. So what I'd like to bring your attention to here is the defocus. This essentially is how far away from the original position that we actually are. And we have MF here. This is manual focus, and we're able to do a large positive or large negative step. This would be a 1 micron step, and we're going to do a 0.1 micron step change here. I'm just going to use 1 micron. Hopefully you can see the 1 positive change. Hopefully you can see that the values are actually changing down here and up here. I've done 2 steps, so a defocus value of 2. And I'm just going to go for 1 more to make it 4 steps. Now, hopefully now, I think it's become quite clear. We've got some interesting objects in the image. We have a VLP here, a couple of them here. They have a bright edge and dark center. And then we've got some other debris and particle matter that may just be carry on from the purification process. But really what we're actually going to be interested in are these particular particles here. What I'll actually do is take a quick snapshot, and then I'll just highlight to you, just in case it's not clear, what VLPs I'm referring to. So just this little icon here, we have a capture image. So what this will do, we'll take a little snap of the image. I can then click on this. We can now see our image here basically as a still. But over here on the right, we can see roughly where in the grid we actually are. I can actually now zoom in on these images. I'm hoping this is following with you. We've actually got some measuring tools here, and we've been informed by Noel, who kindly donated these samples, that on average we should be looking at around 40 microns for the diameter of a VLP, and that's pretty much where we are. Around 37 there was quite close. You can take a look at another one just here. Just apply a measuring tool, around 35. And then another one here as well, 38. So these are definitely our VLPs that we're interested in. We've got plenty plenty to look at here. We've also got some other possible particles that are interesting here, maybe some subunits or some other particles that may have broken off from the VLPs. These are much smaller. Again, these are around 15 to 17 microns. And later today, we'll actually see if we can actually identify these in the image as well. So this looks like quite a positive region that we can look at. So over here, we can actually assign a waypoint. So this is informal the microscope that this is one of our areas of interest, and we actually like to go here and actually collect some images. I'm just going to move to a new region as well. So I'd like to drop at least three waypoints. So I'm just going to move back up, lower in magnification. I'm just going to adjust the illumination just to protect the sample. And I'm just going to move back up to a lower magnification and higher field of view. Okay. So we can start, let's try and start to look for a fresh region. Maybe somewhere a little bit to the right. This actually look not too bad as well. Staining looks quite even and nice. Okay, so again, what I'll be able to do now, I'm just going to move back to the 1 micron field of view. That seemed like a good magnification for these particles. Just allow the system to catch up. Again, we're going to decrease that illumination value to enhance the image. Okay, objects look a little bit blurry at the moment. That's fine. What we can do now, we can use the autofocus again and allow that to run. And pretty instantly, it kind of knows where it needs to be, but it will still go through the initial 10 micron steps, followed by the 2 micron, followed by 1 micron to try and find the optimal focus position. We do have a couple of that we can actually use to assist in finding the focus. We do have a tool called Focus Wobbler, and essentially what this does is tilt the beam left and right, side to side, and this essentially, once the optimal position has been found, the image should no longer shift. So while the Focus Wobbler is active, you can move up and down in the Z plane, and once the image stops shifting, again you found your optimal position. You may need to defocus slightly just to enhance the contrast around the edges of the object. But again, it's a process that can be done just to quickly find the optimal focus, and then again set a waypoint, and then allow the system to then capture images at that given position. So the autofocus appears to be getting quite close. A couple of more little steps, and then I think we should be there. Great, now that's just about done. Again, it's a little bit out of focus for my eye and how I feel the image analysis algorithms will work. So again, I'll just use the manual focus here just to move up 1 step at a time, just so that the defocus value changes 1 step with me. That's 2, and I think again 4 seems to be a good position. Somewhere around there, and again you can see a number of VLPs are present, some other debris and particles as well, but that's fine. Some slight grouping here of 2 VLPs, but I think that's pretty good. Quite interested in this area again, so I'm going to drop another waypoint. Maybe we'll probably move away from around here. Maybe we'll take a bit of a look now somewhere below the center. So I'll just again adjust the illumination. We'll go back out to our 23 micron field of view, just to give us a high level view of where we are. Just So allow the stage to catch up, and then we can now move along. Slight tear there, but that's fine. Okay, this looks another quite interesting region. Now we'll just move down to 1 micron field of view again. I think this will be the last waypoint that I'll drop, and then we can move on to how you can actually capture images at these waypoints in an automatic fashion. So I'm just going to run the autofocus again. So typically, when using the MiniTEM particle analysis, we typically look for around 4 to 5 waypoints and collecting around 25 images per waypoint to get a good representation. Obviously for the purposes of today's demo and the time restraints, I think three waypoints and around 12 images per waypoint should be sufficient. That then allow us to perform some analysis on those particular particles. And whilst the MiniTEM is actually collecting the images, I'll just change back into a presentation just to introduce some of the image analysis methods that we have with the MiniTEM. And I'll also spend a bit more time talking about our machine learning approach to analyzing VLPs through convolutional neural networks. Just give you a brief introduction of what these are, how we've been using them, and what we hope will be the positive impact they can have in characterizing some more complex particle types. So we'll just wait for the autofocus to finish. Need to get quite close now. So this particular region looks a little bit drier, probably not as optimal as the first two waypoints. So I may not particularly use this region. Doesn't look as good. But we'll see how the focusing looks at the end. Okay. There's not as many particles present in this particular region. So I may quickly try and find another region to drop a third waypoint. So again, you can, if you particularly have a region that you think will be quite interesting, you can actually manually input the stage coordinates in here, hit enter, and the microscope automatically return to that position. So it could be useful if you've, excuse me, you've actually imaged the group before and you want to go back. Each time the MiniTEM actually captures an image, it will actually record its X and Y position. Hopefully this text is very small, but hopefully it will tell you where it is. And providing the grid hasn't been removed, you can always go back to that position and acquire some more images. I think this image looks a little bit more even in terms of staining, so I'm actually going to drop this as a waypoint. So that's my 3 waypoints I'm going to be using now. Be pretty happy with that. So what we do now, we're actually going to ask the MiniTEM to actually acquire images at each of these waypoints and perform autofocusing at each one. So we actually now need to move from our imaging panel, which is up here, to our workflow panel. Okay, so this is a fresh project, so we actually haven't got any workflows already saved. I'm actually going to create a new one and just take you through the process. So in our workflow panel, we actually want to create a brand new workflow. I'm going to call this image capture. Once you click that, it then gives you a little bit more freedom to actually interact with this particular section. The little red asterisk just means that the workflow hasn't been actually saved. But right now we have the option to apply some modules. Now our image processing, basically our workflows consist of a variety of different modules of different types. For the actual collection of images, we'll need to worry about an input on our output. And later I will talk about some of the additional modules when I'm actually working with the CNNs. So if I click on the input, I actually want to perform a process called screening. I can then drag this over into the center. And then we also want to make sure that we actually save our images. So on the output tab, we actually have a process called save image. So you may notice if you actually hover over many of the icons in the MiniTEM software, there's plenty of tooltips to actually tell you what's actually happening as well. And down here, although it may be quite small to see, there's actually a little help icon, and the text will be incredibly small for you, but essentially this will actually give you a bit more information about what each actual module is actually doing. Save image is pretty self-explanatory, but for the more complex ones, there's a bit more information to help guide you in terms of what that module is actually going to do. So first of all, the screening module. There's a couple of different parameters that we just need to work through. We want to use the current magnification. We have this checked because we do want to use the current magnification, which is 1 micron field of view. We do want to use the autofocus function because we're using multiple waypoints, which may have a slightly different Z value. So we want to make sure that we can compensate for that. I found that a defocus value of +4 seemed to work well with the autofocus. So I'm going to input that here. I'm then going to ask it to perform imaging at the waypoints. So we have 3 of those. And I'm going to ask it to do four images by 4 images. So region width just means a 4 by 4, basically grid, as the MiniTEM will essentially raster scan across this particular waypoint and acquire a number of images. Image separation, you can actually control how much separation you actually want between individual images. 110 just means 10%, essentially. It's going to be a 10% separation. So it would be roughly 100 nanometers separation between each image, just to ensure that they don't overlap. And then at the bottom here, we have dark image rejection and dark pixel threshold. So these 2 values actually communicate and require some interaction. Dark pixel threshold is what we can actually essentially assign what the actual pixel value is defined as actually dark. So in this case, it's at 50%, which will be 50% of the actual bit range of the camera. Now, I'm actually using a slightly lower illumination value, so the images may appear slightly darker. So what I'm going to actually do is reduce this down to 10%, because I know naturally the pixel values that I will get will be lower than using the auto illumination. So I want to make sure that the screening module doesn't automatically throw away any images I collect, as I will define them as actually being too dark. So I want to reduce that value. You can even set it at 0, just in case you worry about any images accidentally being discarded. Dark image rejection. This is quite useful in terms of if you accidentally image over a grid bar, which will be completely black. If that takes up more than 50% of the image, the software will automatically discard that image, so you won't immediately get an image that only has 10% of your sample and the rest of it just being a black empty space. I'm going to leave that at 50% just in case we come across any grid bars or any large debris that may be present in the or staining precipitate that may be still in the actual sample. We'll leave that as it is. For pre-validation, I don't think we really need to worry about that. Pre-validation will just essentially show me every single image and ask me if I like it or not. Again, if you're only capturing a couple of images, that may be fine. If you're capturing 10, 20, or even 100 images, it will obviously take quite a bit of time. So we'll leave that unchecked. I'm now going to ask the module to communicate with the save image, so that will automatically save the image after it's been captured. We have an option to say skip if no particles, but we know there's particles present, so we're fine here. I'm going to actually add an image tag. So I'm going to put VLP waypoint for waypoints. No need to save the analysis, but essentially with the save analysis and target analysis layer, you can actually automatically capture images and then go straight into an image analysis workflow or pipeline. We're not doing that at the moment, but again, if you have something optimized, you could basically just acquire the images and directly perform image analysis on those images, and it will automatically be saved. You can actually streamline the process a little bit more. So I'm pretty happy with these parameters. So what I'll do now is I'll actually click to run the workflow, and it will begin the autofocusing process at the first waypoint. So hopefully what you'll see here, eventually you'll actually start to see our images populating this preview page, which will actually, put that back there, sorry. And we'll actually see our images being collected, and then we'll also have a log here. So the log will actually tell you everything that the MiniTEM is actually doing at the moment. So at the moment, it was moving to region 1, or the waypoint. What it will then do is then perform autofocusing, which it is now. This process will take roughly 90 seconds, as we've shown earlier. And then it will go through and apply the defocus values and then start capturing the images at this point. So while this is happening, I'm just going to switch to my slides. So I'm just going to stop presenting at the moment and then switch to another one. Please bear with me 1 second. Okay, so hopefully now you can see my presentation.
We can see your presentation.
Great, thank you. So of course, now while the MiniTEM is busy collecting images, I just want to introduce image analysis options for TEM images, and also our machine learning options that we have, which are convolutional neural networks, or CNNs for short. And then also discuss how TEM can actually be used as an orthogonal technique for some of the gold standard analytics that are present for the vaccine manufacturing as well. So hopefully you can see on this slide just some example images of viral particles. And obviously generating the images is actually one of the easier parts. But when you've got 10, 20, or even hundreds, or possibly even thousands of images, it can become a potential bottleneck when you're actually trying to extract some meaningful data from these images, so that you can inform upon your sample in terms of its actual quality and condition. And there's many ways that we can actually try to achieve this. You may do it manually, which obviously will be very, very time consuming. You have many hundreds of images. You may use some of the open source tools that are available to you, such as ImageJ or Fiji. And of course, we have our own options that I'll be presenting in the next slide. But ultimately, the goal is the same. We want to be able to extract meaningful data from these images so we can best inform upon our sample, make the best decisions of our preparation and purification steps. And the best way to do this is in some form of automated fashion, because essentially it will save a lot of time. It can become more standardized, and we can have a bit more control over it as it becomes less subjective. So for the MiniTEM, we have two primary ways of actually extracting data from our images. The first is our workflow modules. Briefly, we'll have just saw that on the actual MiniTEM itself, where we set up the image screening. But essentially, we have different tools and modules. So essentially, we can capture images, we can select images, we can then transform those images to try and enhance the edges, which will then allow us to detect the particles themselves. And then we can then apply different filters and classifications to then extract out different particle types. So you may have an AAV particle, for example, you may have a cluster or debris. Now, these can be set up in an automated way that can just process images in a matter of seconds. And obviously this can help streamline the process. And these work quite well for symmetric particle types. They can become a bit more challenging when you have particle types that are pleomorphic or highly variable in their size and morphology. And this is why we've gone down the route of pursuing some artificial intelligence or machine learning models. Essentially, we've started developing some experimental models for AAVs and also lentiviral vectors, because these tend to have, as shown in the image here, these tend to have quite a variable morphology, variable size and shape. And this corresponds quite nicely for VLPs as well. Depending on their design, then again, they can vary wildly in terms of their size and morphology, and also may present a pleomorphic structure that may be more difficult to detect using our standard workflows. But either way, these can both be actually used in an automated way to generate quick data. It can take a matter of minutes to process a number of images. So just a little bit more about machine learning. Machine learning is quite a popular term at the moment. It's pretty much everywhere. It's already part of our lives and will probably continue to do so, very much so in the near future. But it has a lot of potential for automating image analysis. It's already pretty active in light microscopy, and certainly there's definitely going to be a use in TEM as well. Now, the branch that we will be working with today are convolutional neural networks, and these have been inspired by basically the animal visual cortex. And how they basically work is that we present the model with trained material or classified data. We essentially tell the model that these are the objects that we're interested in. The model will then seek out to try and identify those in a set of image. We present it with lots and lots of images of that, and eventually it trains and becomes more akin to actually be able to actually detect these objects of interest more reliably. We don't actually need to tell the model why we want it, just that we actually are particularly interested in that. And some of the very early tests of these particular models in computer vision as well was done with cats and dogs. And obviously it works quite well. They have different sizes, different shapes, different textures, different colors. And it's pretty straightforward for a machine learning model now to distinguish between these particular image types or even animals. It can become a bit more complex when you start working with objects that have a similar shape, so apples and oranges, but we can still see that there's differences in color. There's definitely differences in texture as well. And it becomes a little bit more challenging again when you start actually looking at viral particles and VLPs. Color is kind of removed from that. We're all working in grayscale, but you can actually look at different types of viral particles will have different possible texture features as well. An example of rotavirus here. And again, there may be slightly differences in size. But if a model's been presented with enough examples of a particular image, have been shown what to actually detect for, over time it can faithfully identify these particular different particle types, which is where we're actually very interested in. Now, our baseline CNN model has been trained on many thousands of TEM images and has been annotated by our experts in-house in Stockholm. So this baseline model is at the stage now where it only needs to see a handful of new images to actually branch off and create a separate CNN model. So in this case, we have rotavirus in this particular image, but it would only need to see a certain number of AAVs or VLPs to quickly learn how to identify these particular particle types. So what this allows us to do is expand the applicability of these models. And this is why we're actually going to be looking at this in terms of how we can expand that towards VLPs. So now it's just a little scheme just to show you how the very early stages of how you can actually train a CNN model. There's a bit more detail that goes into it. This is essentially how it works at the very first stage. We start with our TEM image. We then start to annotate those images. We start to identify what our primary and secondary and tertiary particles may be. Maybe a viral particle, it may be a cluster, it may be an aggregate. We then repeat this process for a number of images. And then we then feed this into the actual CNN model itself. And the CNN model will then jostle to try and find the best fit between its input or training data and its predicted image outcome. Once it can make no further improvements after we've been monitoring its progression, we'll then stop and save that particular model and then test it on a validation data set. It's then that we hope then that it will then faithfully detect are particles that we're actually interested in. And when we spread this out across a number of images, you know, hopefully generate some interesting and powerful metrics from these. So in a nutshell, that's how our CNNs work. I'm just going to show some example images before moving back to the MiniTEM microscope. So what you're seeing here is an image of AAVs. And typically what we're interested in here is identifying whether these particles are internally stained, which is an example here is a dark centered cell, where the stain is actually either inside or on top of the virus. And then non-internally stained that have a bright center. So when I actually overlay the segmentation and particle characterization of the CNN, you can actually see that it performs very well. In green are our non-internally stained, and then in red we actually have our internally stained. And this particular example is very much in favor of non-internally stained. And this is quite an ideal sample. There's very little background. There's very little aggregation as well, and all the particles are around a similar size, so it's quite easy for the model to perform. It can become a bit more challenging when you start looking at VLPs, which we are today. And hopefully you can see the red arrows, and that will indicate where the VLPs are. But then there's also other things that are actually present in this image. We've got some large aggregates or debris that are present, possibly from the purification process or the cell lysis. And we also have some smaller objects as well. And we need to distinguish whether these are actually important to us, whether we want to quantify them, or we just want to ignore them. We just need to know whether we actually want to change our purification process to try and filter these out. But really, we're going to be interested in the VLPs themselves. So as mentioned earlier by Salome, we're actually going to look at some images that were kindly provided by Kirsty, McKenna, and Gibson. As you can see, rotavirus present here. We've also obviously rotavirus up here as well. We've got some latex beads that are present, and very, very small viral subunits as well. It'd be very difficult for you to see possibly, but I will zoom in the following slide. So we've actually been experimenting and using the CNN to try and detect these 3 different particle types. So hopefully when I overlay the segmentation, you can see in green, we can identify our viruses. And then in blue, we can identify our latex beads. And then in a purple color, we can identify our viral subunits as well. Is that a little bit bigger for you? Just so you can clearly see, because we've got our viral particles, our subunits, and an example of the latex beads as well. So this is quite a complex sample with a lot going on. And hopefully this gives you an idea of the potential of the CNNs to actually automatically extract the data from these images, which would be otherwise quite tricky to do with some of the existing or traditional image processing that can be done at the moment. Then finally, before moving back to the system, it's just what can TEM actually help in comparison to complement some of the existing methods that we may use, be that digital PCR, analytical ultracentrifugation, or dynamic light scattering as well. TEM actually allows you to actually visually inspect your sample, and that's interesting in itself, but they can also then allow us to observe whether there's any debris present, such as proteasomes, as the given example here. We can see if there's aggregation of our samples. Then also the actual integrity of our samples as well. We can determine whether actually they've been fractured or broken during any one of our processes to actually try and purify our samples. This is all important information. So when we're actually trying to optimize our processes, we may use different types of filtering and purification steps. In the particular example here on the left, we're looking at the baculovirus helper system. And you can see when using affinity chromatography, actually the baculovirus doesn't actually get filtered out. It's still actually present, which is not ideal. But when actually using a sucrose gradient, you can actually see that the baculovirus is actually removed, and we just have the AAV particles that remain. This is something that TEM can actually offer. And when we're actually looking at our formulation buffers, you can actually see if the change or the composition of the buffer may lead to particles aggregating or being slightly damaged in the process. So again, it's all valuable information going to help you confirm and validate some of your findings using some of the existing technologies that are used for analytics in vaccine manufacturing. I've mentioned a bit more about viral particles rather than VLPs. So I just wanted to quickly mention some of our ongoing and upcoming projects that we've been working on. So currently ongoing, we've currently been supported by Vax-Hub for the interaction voucher, MiniTEM analysis for characterization of viral vaccines, vectors, and VLPs. This is working with UCL and ourselves. The goal here is actually we're going to look to optimize sample preparation using non-radioactive negative staining, such as nanovan and nano-W, just to see the applicability of these and how they compare to the gold standard urinal acetate. And then we're also going to be looking to implement and optimize our image processing for characterization of these different particle types. Then leading on from that, we have the feasibility study, which is going to be a collaboration between UCL and also the NPL as well as ourselves. We're really going to be looking to validate the MiniTEM as an analytical quality control tool. And there's going to be a heavy focus on VLPs and that. We're going to be using the expertise at NPL for generation of VLPs, and also their vast experience in method validation as well. UCL will be sharing their collaborations in low or middle income countries, just to show how hopefully the MiniTEM, our image analysis and CNN can actually be of benefit to those particular regions and countries. Okay, right, now hopefully we'll switch back to the MiniTEM system and see how we've been getting on. Log back in. It's nearly finished. We're quite close now. Timed that, not too bad. So obviously you can now see there's images that are actually populating in the preview page. These are a little bit dark, not to worry about these. I'm going to make that a little bit bigger for you so you can actually see what's actually happening there. Okay, that looks okay. What we'll do is on the actual images themselves, we can actually adjust the histogram to actually enhance the contrast and make these images look brighter, so they won't always look this dark. We've also got our logs that will essentially be showing us what's been going on with the system, and we can see that it's actually performed its imaging automatically, which is great. And it will give you an up-to-date on what's actually occurring, where it is in terms of its image acquisition, where it's actually acquired the images, and if it's had any issues. So I'm just going to quickly look through here, and I can't see that there's been any particular problems, so, which is great. So what we can actually do now is actually move to our analysis tab. So these images will be automatically saved, and it will be shifted to our analysis tab. So I shall click here. And these are some images that I've automatically imported. So what I'll actually do is I can actually filter. So hopefully what you can see in our filtering here is that we can filter images in a variety of different ways. We can apply image tags, which is what I've just done. The image tag that I applied was VLP waypoints. And I've just got some example images as well in here as well, just to show you a bit more options. And again, we can filter in many different ways. If we have fields of view, we only have one field of view, but if you have multiple fields of view, you can quickly filter your images. We've got 148 images shown up here in the top. So these filters can become very handy when you've got a large data set that you're particularly looking at. So I'm just going to click on our VLP waypoints. This is dark at the moment, but I wouldn't worry. Okay, just click that also, just to adjust that. Just take a look at some images here. Okay, so you can see some representative images. Just make these a bit bigger for you. And we've got our VLPs quite here, which is great, which is good. We've got a lot of other matter that's in here, and that's fine. We don't really need to worry about that at the moment, but it's good. We can see what's going on. We've got a nice visual representation of how the sample is. And that's, this is really nice information. The next challenge is actually going to be to actually manually identify these particles, then hope to perform some particle counts on these, maybe measure their average area. We're actually expecting them to be around 40 nanometers, so hopefully we'll be somewhere around that ballpark. And we do have some tiny little particles like this that I've occasionally seen dotted around, and I will also try and see if I can actually identify these as well. Okay. this looks pretty good. So now that I'm actually going to use the CNN, at the moment the CNNs are, say, they are experimental. They're not a locked program as such. So I just need to switch to a different PC, which will hopefully will not cause any problems. That will allow me to use the CNNs, and then actually we can actually see how that actually looks and actually identifies our particles of interest. So I'm just going to switch to PC. I've also got the password wrong. Brilliant. Okay. I'll close that and restart. So I'm currently sharing a database. So I'm on a separate PC now, and hopefully when I click on the analysis tab, we should have a complete mirroring of the same images, which we do, which is great. So I can then apply the same filter, and then we'll have our images here, which is good. So I'm just going to start initially with 6 or so images. So I'm just going to make that auto so you can actually see. That's fine. And I'm actually just going to set up an image analysis workflow just for these 6 images. So I'm actually going to give these a little tag. So we have a little tag icon up here at the top. We can add a different tag. So I'm going to test CNN on there. We apply that, and now hopefully you can see in the image tags here, we have our original image tag from the image collection, VLP WPs, and actually our additional tag here now, which is test CNN. And I'll use this test CNN tag to actually select the images when I actually want to run the CNN and the CNN module. So what I'll do now is actually just move to the workflow tab. Okay, I'm going to click on CNN test. This is like pre-saved. As you'll see, it's empty. So I'm just going to walk you through how I would actually create a very initial CNN test module. So you want to click on our modules tab. You want to click on our input. We actually want to select our images tab. So we're not actually screening this time. We want to select our images. Just going to drag that over into the main panel. Just move that up. So we have the little filter icon here. I'm going to use the test CNN. We can now see, hopefully, that we're only going to analyze 6 of our 149 images that we have. We don't need to worry about the analysis layer at this stage. So I'm actually going to use the CNN to actually detect. We have our infer CNN here. Let's move that into the center. Collect that up. There's just a couple of parameters that need to be inputted here. So the first thing is I actually want to detect VLPs. I have a class assigned. I'm going to call that VLPs. Resize factor. So when the model is being trained, we actually reduce the size of the image by 50%. So I'm actually going to make sure that that matches up. That is 55. And I'm now just going to apply my detection model. So detection model is going to be my trained models, VLP version 2, and then we have a segmentation model, which will allow us to actually segment the cells, which will be living in our model. So, mask. And this is all we need to do to actually set up a baseline detection. I'm going to make sure that the detection is as tight as possible around the outside of the particles. So I'm actually going to use a tool called Refine Particles. And what this does is actually blur the edge. Well, essentially, it blurs the image ever so slightly, helps enhance the edges of the particles that we're interested in, and it will actually then help the segmentation or identification to be a bit more tight around that. One thing we just need to change here is the refinement geometry. Our objects are not going to be circles, they're going to be slightly irregular or pleomorphic, so we actually want to use the pleomorphic tool on this one. And then finally, we want to save our analysis. So for our output, I'm going to move down to save analysis. So I'll just drop that in there. And we'll just link these up. We can add another image tag as well, but I'll leave that blank for now. And I'll talk about the image analysis layer in a moment. I'm going to ask these to be dropped onto layer 2. Okay, and that is essentially all we need to do to set up our first initial test. So I'm just going to make sure that is saved, and I'm now going to ask the model to run. So hopefully you can see over here on the right, I'll just make these a little bit bigger, is that you'll see the model working through its steps. It's applying the CNN, it then refines the particles and saves the analysis. You may only briefly see that when it actually detects the VLPs, it will flash red, and then it will move on to the next image. You can see it's taking roughly 8 seconds or so to perform. Even less than that, it's actually taking around 2 seconds to perform the analysis. We're already through 4 of the 6 images. And we're almost done on the last one. We can move back to the analysis tab to see how this particularly looks. This is probably the first pass that I would do. I can clearly see from here that we've got some VLP particles or possibly some false detections that look a lot smaller. And essentially you can apply different filters. You can filter these out based on their size and their morphology. There's different tools that we can do. And I will show you a data set that I've already applied this, and you can actually see how this is done. Okay, this is done. We've completed the workflow. We've actually got no issues reported, which is all positive news. We can then move back to the analysis tab. And I'm just going to filter on our test CNNs with our 6 images that we're interested in. Now you may remember the layers. So if you're familiar with any Illustrator software, such as Photoshop, you can apply different layers and different layers to your image. This is very similar here, is that we can apply different analysis layers. So we've got an option of up to 5. Layer 1 is I've left this completely empty, just so we can see the natural unanalyzed image. But on layer 2 is where we actually perform the particle detection. And obviously 3, 4, and 5 have been left blank. So this gives us some options if you want to actually try a different set of analysis. We can actually then apply it to a different analysis layer, and then we can compare between these to see which one works the best. So if we're just looking here, again, we can just see in purple, hopefully you can see some detection. And as a first pass, it's worked pretty well. Possibly this particle here may have been too small, which is possible. I can actually switch off the segmentation just to see, and I'd say it probably is a little bit too small. And again, this one may or may not be a VLP. It's got a bit of a dark center. It certainly does seem to be potentially a VLP or the beginnings of one. So I'll leave that one as it is. You kind of can see it's missed some of the larger particle matter that are in there, which is good. We don't want it to detect it at this stage, but it's worked pretty well. You can check through the images, switch the segmentation on and off. You can see it's worked well again. Anything that's on the edges will automatically be excluded by the software. You can see there's a little bit of a false detection down here, such as area. So you may want to actually go back and filter some of these out. It's worked quite well. Again, this one again might be too big or irregular shape. You can filter these. But as a first pass, it's worked pretty well in automatically detecting these particular particles. So it looks pretty good. So what I may quickly do is just apply a small filter. Actually, what I'll do, I'll show you first is what you can actually do. You can actually click on plot here. We can then plot this blade. Now at the moment we've only got 4 particles present in the image, which is fine, and we'll analyze more images to actually increase that data set. But what you can actually do here is actually plot histograms. You can plot bar plots. You can also plot scatter plots as well, where you compare possibly the circularity against the area. We can change what we label on the X and Y axis. We can use different parameters of the actual objects to plot against each other to see different trends. But what I'm actually interested in is kind of the size of these particles. So if I go on plot of particles, we actually see a tabular form of the actual information. And I actually see here that we've got an area of around 12,000 nanometers squared and 875. So we had some of these smaller objects that were present. So I'll be interested to see how big these were. So this particular, if I click on that particular particle, you can see this is the image, it gets highlighted. So the area here was around 400, and I think that's a little bit too small. Click on the, we'll filter these by size. So click on the largest object. That looks around, it's around the right size, I'd say. And we were interested in the area, actually, what was the diameter of these particles? As you can see, it's quite close to the 40 nanometer range, which is what we were actually aiming for, which is great. So what I'm going to do is I'm going to apply a filter for size. I'm going to do slightly larger than this, maybe do 1600, and I'm going to set a lower end of around, click on that particle, 800. So I think anything below 800, anything above 1500, we can then filter these out. So I'll just apply a quick filter in here, just move that down. So where we're interested in now is measurement. Click on measurement, we have an option to use a particle filter. You can drop this in here, link this up, like so. And I think we said that we wanted it to be anywhere around 800. And we can maybe put something like 1600 here in terms of nanometer squared area. And we're only interested in particles that actually pass this filter. Anything that is blocked, we're not interested in at this stage. So I'm just going to ask this to be passed on to the image and the analysis as well. And I'll change the analysis layer to 3, so we can actually compare between 2 and 3. What I'll actually do is ask that to run again. This shouldn't take too long, but what hopefully you'll see in the log file is that when the filter is applied, you'll see that it will actually potentially remove particles. So as here, particle filter has removed 2 of 8 particles. So we're just moving through now image 3 of 6. It's working quite well. So some of these irregular objects may still come through depending on their size, and you could apply another filter for potentially elongatedness. So obviously any object that's more crescent shaped will have a higher elongated value than the VLPs that are going to be more circular. Lots of different things we can do to build up the complexity of these CNNs to ultimately get where we need to get. It's almost done. We can move back to the analysis tab. Hopefully, where we had some of the smaller objects, we can actually filter these out. So if I toggle between image layer 2 and 3, you can see that we start to lose some of these smaller objects, which is exactly what we actually wanted to do. Trying to find an example again of the smaller particles, possibly here and up here. You can see that they've actually been. So you can see that again, some of these objects here that actually gets filtered out. So if we wanted to actually combine all these images together and actually plot some data, what we can do is actually we can click Control A, we select all these, we move back to the plot particles feature, and we actually want to say plot filtered. So that will quickly calculate the metrics from these 6 images. And then we can actually see a plot of area against circularity as well, which is good. If we can move to a bar plot. Again, we've only got one particle type at the moment, VLPs, so there's only one bar. But if we do move to a more complex model where we're looking at different particle types, you can start to break this down into different types. So this is what I'll actually show you now. So what we can do. So we'll move to a more complex CNN. Just make this a little bit bigger for you so you can see. Okay, this is similar to what we've been working on. So we've actually got our VLP CNN. We've then refined our particles and then we've filtered. We made this a little bit more interesting. Anything that passes the filter, which is the VLPs that we want, this is passed on. Anything that is blocked, we've defined as other. So hopefully what this will do, this will then flag to you and say, this is outside my filter. What is it? It could be a debris, it could be, or it could be a doublet of VLPs, because it would be outside the maximum area value. So then this will then bring it to your attention. You can then go and look at it and say that's a doublet, possibly assign that as a different class. Over on this side, I've actually looked to identify objects that look a little bit like viral subunits. May not actually be subunits, but there's a close similarity. Actually asked a second CNN model to work with the first, and then filter that out based on size, and then this is all brought together into one image, and then it's actually saved. So hopefully what I can actually show you now is what that would actually look like. So I'm just going to show you these particular images. So load up all the example images. So just to show you what we're actually looking at here, these are some of the VLPs again. Let's make this image bigger for you. So this one's fallen out because it's possibly too small. You may want to reclassify that as a VLP, which you can do. You can actually, we'll just plot displayed. You can actually click. This particle is highlighted in the list, and you can assign a different class. So if you actually wanted to assign that, you can move down to our list and say VLP. Now this is now a VLP. Alternatively, we can still leave it where it was as another, as we can. Hopefully in this image you can see VLPs and actually some of these objects that look a little bit like subunits, possibly in there. Can't say it's 100% true, but it is, it's just to show you that you can start to take other objects inside your images should you wish. Try and give some examples. Trying to find a doublet for you. So here's a doublet. It's just about fallen inside the actual image. There's some of these interesting objects here. Again, this has been called as a subunit, but also an other because it's a smaller object. And again, we can apply different parameters, try and exclude these from each other because they're overlapping. But there's a little bit more going on in this image. So actually what I'm going to try and show you now is if you actually plot the data from, say, 2 of these particular data sets, so 50 images, and select all of these. I'm going to plot filtered. Hopefully that will update there. So when things become a little bit more complex, hopefully you can see that you can actually get particle counts for other objects, which is our yellow, our subunits, which is 273, and then finally we've got our VLPs as well. So that's just a straightforward particle count. This can be exported as a white background or transparent background as well. And you can also see this as I think again as a tabular form. You can see all the information corresponding to these particular particles in a list. So we've got some 738 particles in here. You can see all different ones. We can look at their max and minimum diameters of their area, their perimeters, and all different features, circularity and elongatedness as well. Again, this can all be exported in different formats. CSV being the most common, so you can automatically drop that into a statistical analysis package as well. But we were particularly interested to see what the average size of the VLPs were. So we can actually leave that as you go back to a scatter plot. And then you may want to put a diameter around there. So we'll leave that actually as minimum diameter. You can see hopefully you can see a nice plot of what we can actually, the 3 different particle types here as listed below. And we can see that there's some clustering of our VLPs around the 40 nanometer mark, which is indicating that we're detecting them pretty faithfully. I'm pretty happy with that. The subunits are smaller, as to be expected, closer to 20, 22, 23. And then the other will be a wide range because that falls in the lower end of our filter and the upper end of our filter. You may want to actually see that as a box plot as well, of area. You can see the distribution of that. If we just put diameter as well, see the VLP is pretty much around the 40 mark as well. So hopefully this has enabled you to actually see how quickly you can acquire images, apply a CNN to this, and then extract some data from that pretty quickly. The processing of an image may take a couple of seconds. So obviously if you have hundreds of images, it'll take a bit longer. 100 images will roughly take around 30 minutes to generate this amount of data, which is not too bad. So while I'm here now, actually, I'm just going to quickly move to the rotavirus data set, just so you can see something a bit different, a bit more complex as well, and just show you some of the values that you can generate from this as well. So I'm just going to close this project. I'm going to save those changes. Sorry about that. Someone else is actively on the database and they've jumped the queue in terms of my projects. Okay, great. Hopefully make these images a little bit bigger for you. Hopefully I just zoom in, you can take a bit of a look at these particles. So we've got a mixture of rotavirus, latex beads, and we've got viral subunits as well. So these are really quite nice images that have been kindly donated. You can see some debris as well and matter in there, but we're actually not interested in that. And this is how the CNN successfully detects and reports out some of these metrics. And just to give you an idea of how these values work, we've got a lot of samples here. We've got 1 through to 10, which is quite a lot. I'm just going to use 2, just give us 18 images, but there's plenty of actual data in here as well. I'll just zoom that out there, and I'm going to plot filtered. This may take a little bit longer. There's a lot more particles present here, within the many thousands. Give that a bit of a moment. I'll quickly just show you a plot, and then we'll wrap it up and we can take some questions. I was hoping to show you how you actually train the CNN. We may not have any time for that, but just need that to a bar plot. But again, just to show you when we actually start dealing with larger numbers here. So we have 3 different particle types that I've mentioned. Particle type 1 is the rotavirus, particle type 2 is the latex beads, and particle type 3 is viral subunits. And you can see there's obviously there's more subunits present here than there are viruses and beads. We can generate a nice plot for that. So that works pretty nice as well. Then do a scatter plot. You may want to look at area. You get like a nice trend here. You can see there's some variable size in terms of the area of rotavirus, but fairly consistent. Beads are slightly larger, as you can see. And then the subunits are pretty much, obviously very much smaller. And there's variable degree of circularity here, but again, this can give you some nice information of what's present in your sample. Now I'm conscious of time now. I hope it's informative, and we're now happy to take questions. Yes, I'll just switch back and hand over to you, Salome.
Thank you, Adam, for the great presentation. So the Q&A session is now open. Feel free to ask your questions either to Adam or to Saba. No questions? Steve, Adil? Okay, go ahead, Adil.
Thanks, Sal. Adam, great talk. Really, it's nice to see something so complex as EM made simple. But I mean, how much of it do you think is basically getting a good grid preparation? Because garbage in, garbage out, and so how can we use this sort of MiniTEM system to sort of do like a pre-screen for our grids, and then actually before we kind of as a quality control before we actually go about trying to get collect particle information, if that makes sense.
That's a great question, and yeah, the biggest challenge is actually getting consistent sample prep, as you mentioned, which tends to come with with time and practice. It's something we're clearly aware of as well, and we're actually just recently published a short paper that we are working on approaches to automating the sample preparation process, which we know it's a big issue. So hopefully we are trying to think of ways to actually make that possible. What I didn't actually show here as well today is that we are in our upcoming version of the software is that we're actually able to image the entirety of the grid in a way. It's called a grid square imager, and it will actually go around and actually take pretty much image every single mesh on the grid to give you a nice representation of actually what's actually going on. And once you actually find an area that is, which is good, you can actually inform the software to look for these particular regions. So that will take some of the guesswork out of it. You may have seen me just navigating around the grid, trying to find like good areas, and there's some bad and some not so good. But that will help automate that process and make that decision for you, and it will then fixate and then go on to those images and actually image them. I hope that does answer your question in some way.
Yeah, no, absolutely.
Good. It's the biggest issue is the consistent sample prep, and it's the same for the CNN training. You said it. Rubbish in, rubbish out. Because the good quality needs to go in to get good quality out.
I mean, that's really good. I mean, especially the two things that I worry about is just one. These grids, they're very tiny, but when you're looking through the EM, it's sort of navigating several football fields. So you kind of wonder whether if you're on the right pitch or in the right even area to kind of have the better. So there's sometimes you have a horrible grid, but then there's one particular part which looks just amazing, and actually you can get a lot of your information out of that position. So if there was a way for the MiniTEM to do that, so it sounds like the grid, checker is the way forward with that.
Yes, certainly.
Okay, thanks a lot. I'll hand it over if anyone else has any questions. Otherwise, I can keep asking questions.
Can I also just have one more comment on that, Adil?
Yeah, sure. Go ahead.
And Adam, one of the things that I think maybe you guys saw is that some of the detections of those VLPs were done in sort of dark areas. So that this is one of the sort of benefits of using CNN is that when we are training it, we can also give it, not just the perfectly contrasted kind of particles. So some of the ones that were detected were in darker areas, and it could pick that out. If you were just doing a standard workflow when you're sort of standard image analysis, it would lose those particles where it would be very dark in the background, or they would be part of something else. And that should also hopefully help you to pick those particles out that you wouldn't be able to do normally. And as Adam said, this grid square imager is going to be really useful because you can predefine what kind of area you want to look at and then let it go and find those for you.
Fantastic.
Anyone else?
Should I follow this up with sort of the other $1 million question, which is, so when working with really nice particles, VLPs are really, easy to spot, right? You'd think, but sometimes we're working with sort of experimental constructs, and as you said, there's some pleomorphy to it. There's some, but that looks very much like cell debris. I mean, so I've had preps where I've thought, oh, there's a beautiful looking particle, and it's turned out to be maybe membranous sort of vesicle debris or some other kind of aggregate. So, I mean, how well do you think that your CNN systems are able to differentiate, and it is all about this training, isn't it? But I mean, if we're not sure, then basically we have a problem training those networks. So do you guys have any tips or hints as to how we can, improve our shot selection, if you like?
Yeah, that's a tough one. I think if the biggest challenge is can you reliably identify those particles yourself, because that's what ultimately the CNN is going to rely on our input to actually say this is what I'm interested in, this is what I'm not interested in. Just as an example, I was trying to identify some subunit like particles in those last images. There wasn't many of them around, basically. So in terms of the input data that you're able to provide the CNN with, you probably just need you need to ensure that you have confidently have enough of those particles present in a set of images. You may need to take lots of images to actually generate that data, and then you'd hope then it would actually be pretty faithful in terms of how it actually identifies those particles. Again, it's always going to be tricky if the particle that you're really interested in looks a lot like debris or some other membranous matter. So I can't give a definitive answer. It's one of those we'd have to just try. And but in terms of actually training it, it's not hard. In terms of it's hard, it's just having the quality to put into it, and then it works. It works pretty well.
I guess it's sort of trying to get your grid, a good representation on your grid. So, what kind of concentrations would you recommend?
I think for viral particles, we're typically looking at 10 to the 9, 10 to the 13. It's not always possible to do that. I'm aware of that. Obviously, the more the better. But obviously, you have to take into factor the size of the particles. That was for AAVs, for example. You need less for that if the VLPs are particularly bigger. But it's not the more the better, but plenty is the thing. Is plenty. But also when training them as well, you actually want to make what you actually tell the CNN to exclude is just as important as what you include as well. So you still want to have areas of background. So you can actually say this is background, this is not particle, don't detect that. If you've got some membranous structures or debris that you don't want, again, you need to have some of that present as well to actually to show, not to exclude. So what you put in and what you want in and what you want out is just as important. But yeah, I think we're looking at 10 to the 9 particles, I'd say, as a baseline. Per mil, that is.
Yeah, and I can maybe add as well that, I mean, we at Vironova, we've got quite a lot of experience in looking at these kinds of particles. And we always love to sit down with collaborators or clients and kind of share that, because I think we all face these questions of what is it that I'm looking at? Potentially like doing something like gold labeling in the beginning and using other orthogonal methods to target down, like, what are you actually looking at would be quite helpful, at least for the initial identification, so that as well that what you are sort of labeling as your particle is indeed your particle, and then you don't have to do it each time. You don't need to do the gold labeling each time, but it could be a good thing to do in the beginning to know that indeed this is what I'm looking at. And I know they do that often, for example, for exosomes, but it is quite a helpful tool to have. So yeah, it's sort of sitting together, looking at the particles, using other orthogonal methods, seeing what is in your sample from different angles, and then trying to kind of narrow down with confidence what you're actually looking at, especially when it comes to VLPs.
That's a really good suggestion. Yeah, thanks a lot.
Yeah, I have a question as well. So obviously the concentration is important, but do you see any dependence or eye interference when we change the type of filtration that we are using on our samples? The purification, I mean.
Yeah, definitely. The purification methods can definitely have an impact in what actually passes and what doesn't passes, and likewise that may actually impact the particles themselves. It'd always be nice if you can actually compare different methods and then obviously image the grid so you can see how that's actually having an effect on your samples, whether you're removing some of the debris that you're not interested in, whilst leaving your particles intact and largely intact in terms of they're not damaged. Hopefully that half answers your question.
Yeah.
And also to not be afraid of bringing samples from before a step. You can do a quick check, and often maybe there is a lot of contamination that can obscure your sample, but it is always nice to see what happens before and what happens after, because it could be that during a purification step, let's say if you're doing ion exchange, then the low pH, or if you have high salt or something, can have a different effect on your particles, and it will be nice to kind of compare and see, well, what happened before. So you might have a dirtier sample before, but your particle integrity was potentially better, and then you do your ion exchange or whatever, and then the sample is cleaner, but your particle isn't very happy. And I think that's also important information to kind of carry along with you, to kind of go and see it before and after, even though you might not get really nice, like data that you can confidently quantify. Just even that visual observation and looking at it as you would do with a light microscope, I think would be quite helpful.
Okay, thanks. Adil?
Yeah, I just remember one other question, but I just to say, yeah, I agree with you, Saba. I think that's really, I mean, back in the day when I used to have a microscope almost exclusively to myself, I used to be able to sort of do a purification and go and, instead of running a gel, I would just pop onto the microscope and absolutely, you know, where particles are working, it's a really great way and a quick way to sort of just be sure you're looking at the right fractions. My other question was, you know, so we already have done EM, let's say, and you've got images. Can we use our raw TIFF files with the Vironova software and, you know, your neural networks? Because this is actually something be super cool. You know, I'm looking at particles, but also there's interest in sort of, you know, other amorphous fibrillar type of stuff, which could be interesting. So you know, the reality is that my ImageJ or Fiji or, you know-how is just woeful. I think I'm just not technically gifted with computers. So so I just couldn't. I could just, you know, all this thresholding and all this stuff I was useless at. But so you've already got a package, and that seems to me really useful. You know, even in itself to help kind of look at old data. So is that possible?
Yeah, it is possible. I think we use OME TIFF, so open microscopy environments, because obviously that will contain the necessary metadata for our software to actually read those files. And I think there is a way to actually do that through ImageJ or Fiji to actually give you that data. And then yeah, these can then be imported into our software and then looked at. So the rotavirus particles that we showed, that was, you know, they weren't generated using the MiniTEM. They were generated on a different microscope. So it is completely possible. And then we could try an experimental CNN on them and see what we can get. And we're interested in pushing the boundaries of the CNNs and see what they can actually do. So that'd be something that'd be very interesting.
Oh, that's great. I mean, if you could share that, you know, how that technical note on how to sort of convert the files, then that would be useful.
Sure. Good.
Thank you.
Yeah, do get in touch with us, Adil. That would be great.
Thanks, I will do.
Any more questions? Well, if not, I will end this session. I want to thank you again to Adam for the great presentation and to Vironova in general. Also to Noel and to Christy for kindly provide us with the samples for this webinar. And see you next time.
Yeah, great. Thank you, everyone. Thanks for taking time today.
Thank you, everyone.
Thank you. Bye-bye.